Everyone, I am Dr. C.S. Glucuronic, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to this channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with summary of shunt pathway. In today's session, we will start with the same. Then we have physiological significance of HMP shunt pathway, in which we will see oxidative pathway is seen in certain tissues, whereas non-oxidative pathway is present in all tissues. Then we have clinical significance of this pathway, in which the most common enzyme deficiency is glucose 6-phosphate dehydrogenase. Then we have polyol pathway of glucose, in which sorbitol, galactitol, and fructose play a role. Good morning, everyone. Let us start talking. HMP shunt pathway. Fructose 6-phosphate is converted to glucose 6-phosphate in the presence of enzyme phosphohexose isomerase, which is nothing but reversal of glycolysis. In this pathway, 5 glucose molecules are generated, 6 glucose carbon dioxide molecules are eliminated, 12 NADPH molecules are generated, and 1 glucose is completely oxidized. The pathway is operating in following organs: liver, adipose tissue, adrenal cortex, mammary glands, testes and ovaries, RBCs, and lens of eye. The oxidative pathway phase of the pathway is seen in the above organs, where NADPH generation is required for lipid synthesis or steroid synthesis. Whereas the non-oxidative phase is present in all tissues, and so synthesis of ribose is possible only in the body. The next point is generation of reducing equivalents. The major metabolic role of the pathway is to provide cytoplasmic NADPH for reductive biosynthesis of fatty acids, cholesterol, and steroids. Next point is free radical scavenging. Free radicals, that is superoxide and hydrogen peroxide, are continuously produced in all cells. This will destroy the DNA, proteins, fatty acids, and biomolecules, and in turn, cells are destroyed. The free radicals are inactivated by some enzymes containing superoxide dismutase, peroxidase, and glutathione reductase. Reduced glutathione reductase is regenerated with the help of NADPH. In the diagram, we can see that certain enzyme systems inactivate the reactive oxygen species. These are superoxide dismutase, peroxidase, and glutathione reductase. Glucose 6-phosphate dehydrogenase, with the help of NADPH, generates glutathione reductase and helps in the reaction. Next point is prevention of methemoglobinemia. NADPH is also required to keep the iron of hemoglobin in the reduced form, that is the ferrous state, and to prevent the accumulation of methemoglobin. Methemoglobin cannot carry oxygen. Next point is detoxification of drugs. Most of the drugs and other foreign substances are detoxified by the liver microsomal P450 enzymes with the help of NADPH. The next point is lens of eye. The maximum concentration of NADPH is seen in lens of eye. NADPH is required for preserving the transparency of the lens. Eighth point is macrophage bactericidal activity. NADPH is required for production of reactive oxygen species by macrophages to kill the bacteria. The next point is availability of ribose. Ribose and deoxyribose are required for DNA and RNA synthesis. In this diagram, we can see generation of reactive oxygen species in macrophages, where macrophages and polymorphonucleosides undergo respiratory bursts and produce oxygen. This oxygen is converted into superoxide with the help of NADPH oxidase. This superoxide undergoes superoxide dismutase reaction to form H2O2. This again under the enzyme myeloperoxidase forms HOCl and it kills the bacteria. Ribose is also necessary for nucleotide and coenzymes. Reversal of non-oxidative phase is present in all tissues by which ribose could be made available. The next point is about ATP. ATP is neither utilized nor produced by the enzyme shunt pathway, HMP shunt pathway. Cells do not use the shunt pathway for energy production. Coming to the next point, clinical significance of shunt pathway. First, GPD deficiency. The enzyme glucose 6 phosphate dehydrogenase may be deficient in some persons. It is the most common enzyme deficiency in clinical practice. The defect is transmitted as X-linked recessive trait. It will lead to drug-induced hemolytic anemia. The deficiency is manifested only when exposed to certain drugs or toxins. Example, intake of antimalarial drugs like primaquine. Primaquine simulates peroxide formation inside RBCs. That in GPD or G6PD deficient cells, the level of NADPH is low. Hence, further production of peroxides will lead to cell lysis. Similarly, ingestion of toxic glycolides present in fava beans may have similar effect. This is also called favism. Sulfa drugs and furanantine may also precipitate the hemolysis, and this will lead to jaundice and severe anemia. The geographical distribution of G6PD deficiency correlates with the malarial endemicity. The enzyme deficiency offers resistance to Plasmodium infection. The parasite requires reduced glutathione for its survival, which will not be available in adequate amounts in the deficiency of GPD. GPD deficiency is reported from all states of India. Next point is methemoglobinemia. G6PD deficient persons will show increased methemoglobin in circulation, even though cyanosis may not be manifested. Third point is thymine deficiency. The transketolase reaction is measured in RBCs as an index of thymine status of an individual. The occurrence and manifestation of Wernicke's-Korsakoff syndrome, that is an encephalopathy, which is seen in alcoholics and those with thymine deficiency, is due to a genetic defect in the enzyme transketolase. Coming to the next cycle of glucose, glucuronic acid pathway. UDP glucose is converted to UDP glucuronic acid in the presence of enzyme UDP-G dehydrogenase and NAD plus to NADH. Then this UDP glucuronic acid is then converted into D-glucuronic acid in the enzyme in the presence of enzyme UDP glucuronidase and removal of UDP. Then D-glucuronic acid is converted into L-uronic acid in the presence of glucuronate reductase and NADPH to NADP+. This L-uronic acid has two pathways. In the first pathway, L-uronic acid is changed to L-uronolactone with the removal of one water molecule. Then this L-uronolactone is transferred by 2-keto L-uronolactone with the enzyme uronolactone oxidase, which is blocked in primates. Then 2-keto L-uronolactone is further changed to ascorbic acid in the lower animals. On the other hand, L-uronic acid is converted to 3-keto L-uronate with the help of enzyme L-glucuronate oxidase and conversion of NADP plus to LADPH. Then this 3-keto L-glucuronate spontaneously changes to L-xylulose with the elimination of carbon dioxide. This L-xylulose is then converted into xylitol with the help of enzyme xylitol dehydrogenase and the NADPH to NADP plus. Then the xylitol is then converted into D-xylulose with the help of NAD plus to NADH plus, NADH plus H plus with the help of enzyme xylulose reductase. Then this xylulose reductase is converted into D-xylulose 5-phosphate and it is later sent to pentose phosphate pathway. Coming to the importance of the glucuronic acid pathway, it provides UDP glucuronic acid, which is active in the form of glucuronic acid. It is used for the following purposes. First, conjugation of bilirubin. Second, conjugation of steroids. Third, conjugation of various drugs, which will make them more water soluble and more easily excretable. And then fourth, synthesis of glycosaminoglycans or GAG. Coming to the effect of drugs, barbiturates, antipyrin, and aminopyrin will increase the uronic acid pathway, leading to availability of more glucuronate for conjugation purpose. Coming to vitamin C in lower animals, the enzyme L-guanolactone oxidase is absent in human beings, primates, guinea pigs, and bats. Hence, ascorbic acid cannot be synthesized by these organisms. Hence, ascorbic acid is an essential nutrient in the diet of human beings. Coming to essential pentosuria, it is one of the members of Carrot tetrad. The incidence is 1 in 2500 births. It is an inborn error of metabolism. In the pathway, L-xylulose is converted to D-xylulose by two enzymes, xylitol dehydrogenase and xylulose reductase. Absence of any of these enzymes leads to pentosuria. In the diagram, we can see L-xylulose is converted into xylitol with the help of enzyme xylitol dehydrogenase. Then xylitol is converted to D-xylulose with the help of enzyme xylulose reductase. L-xylulose is excreted in urine and gives a positive Benedict's test. Barbiturates, aminopyrins, etc. will induce uronic acid pathway and will increase xylulose urea in such patients. This condition does not produce any harm, but it should be differentiated from diabetes mellitus. Coming to the next point, next cycle of glucose pathway, that is polyol pathway of glucose. Sorbitol is very poorly absorbed from intestine. It involves the reduction of glucose by aldose reductase to sorbitol, which can then be oxidized to fructose. This would amount to the interconversion of glucose to fructose. Glucose, when converted to sorbitol, cannot diffuse out of the cell easily and gets trapped there. In this diagram, we can see glucose is converted into sorbitol with the help of enzyme aldose reductase. Then this sorbitol is then converted into fructose with the help of enzyme sorbitol dehydrogenase. Sorbitol is normally present in lens, but in diabetes mellitus, when glucose level is high, the sorbitol concentration also increases in the lens. This leads to osmotic damage of the tissue and development of cataract. Caractitol also causes cataract. Fructose is present in semen in large quantities. It is produced by polyol pathway. The polyol pathway is active in brain and fructose is seen in CSF. This pathway is inactive in liver. Here ends our session. Kindly like, share and subscribe the channel. Thank you.